In this class, we start our lecture of attached growth reactors, which utilize biofilm to perform various biochemical reactions. Almost all microorganisms cluster together in aggregates or in flocks. Certain microbial communities are capable of adhering to surfaces forming biofilm so that they can stay within a desirable environment despite external forces and protect themselves from various environmental stresses. The formation of biofilm is prevalent in nature and in our bodies. They can be harmful or beneficial, or both, depending on our needs. One example of beneficial biofilm is the activated sludge flux that promotes sludge settling. An example of harmful biofilm you may be familiar with is tooth plague. Harmful biofilm can also be found in engineered systems like our drinking water pipelines, shower hoses, medical implants, and multiple industries such as food processing industries, and reduces the efficiency of those systems and even rendering them harmful by causing corrosion, producing harmful chemicals, and causing diseases. Another relevant example of harmful biofilm is a nascent biofilm and associated EPS on the surface of a polyester support fiber on the back of a reverse osmosis membrane. It can increase the transmembrane pressure needed for filtration. It is one of the major causes of membrane fouling. Biofilm in drinking water distribution systems may carry pathogenic microorganisms, posing danger to public health if not managed properly. Biofilm grown on heat exchangers can reduce the heat exchanging efficiency. On the other hand, we can also utilize biofilm in bioreactors to our advantage. For example, they can be used in biofiltration in water purification, in attached growth bioreactors in wastewater treatment, in ethanol production, soil remediation, and used as barriers to contain contaminant plumes. In the context of bioreactors, biomass attaching to surfaces results in long SRT and greatly mitigates the risk of washout. A high biomass concentration can be achieved within the reactor due to biofilm formation, especially if we maximize the biofilm surface area. Additionally, the protection of biofilm also allows biotransformation to occur under unfavorable environmental conditions. To utilize biofilm, we need to be able to model it. In reality, instead of reaching steady states like the suspended biomass, the formation, growth, maturation, and detachment of biofilm is a dynamic process. Moreover, the structure of biofilm is complex and heterogeneous. Microorganisms group together within extracellular polymeric substances, or EPS, on a surface, forming mushroom-like structure. This allows the formation of channels that help optimize mass transport of substrates. This can be very difficult to describe mathematically. We need to simplify it. On a large scale, for example, from the perspective of a whole reactor, we can average out the variations in a dynamic process and assume a dynamic steady state. We can also average out the rough surface of biofilm and assume the biofilm is uniform on the support surface. Therefore, we can condense the 3D biofilm to only one dimension, which is the biofilm depth. On the substratum or support, which we assume is an impermeable and inert material in this class, there is base film with well-defined boundaries. Mass transport within the base film is by molecular diffusion, driven by the concentration gradient created by substrate consumption. On the top of a base film is the surface film, which is quite variable. The relative thickness of base and surface film is defined by age and hydrodynamic characteristics. This picture gives us some concept of the scale that we're looking at. We can further simplify the biofilm into this idealized planar biofilm model on the right. The planar biofilm is uniform in thickness and has a smooth surface. Outside of the surface is a hypothetical boundary layer or stagnant surface layer, in which there's no biomass and no convection. The mass transport in the boundary layer is diffusion. The most simple model assumes only one species in the biofilm with one substrate with a bulk solution concentration of S sub Sb. You can see the substrate concentration profile throughout the depth of the biofilm. It decreases linearly in the boundary layer as there is no reaction, just diffusion. As it reaches the biofilm, it starts to get consumed. The consumption rate decreases as the substrate concentration decreases. The consumption rate decreases to 0 as it reaches the substratum. If the substrate concentration reduces to 0 when it reaches the substratum, we call the biofilm a deep biofilm. If it does not, we call it a shallow biofilm. A key parameter in modeling biofilm is the flux, which is the mass of substrate diffused into a unit surface area of biofilm per time. It is important because the flux is a result of diffusion driven by concentration gradient in the biofilm created by reaction. So the flux should equal to the reaction rate in that unit surface area of biofilm. Flux can be described by Fick's first law. It is the diffusivity times the concentration gradient. Diffusivity essentially indicates how fast the compound travels through a media through diffusion. In the boundary layer, there's no reaction. The substrate concentration gradient is simply S sub Sb minus S sub Ss. In the biofilm layer, the substrate is consumed along the depth of the biofilm. This is analogous of a plug flow reaction. So, we can use the differential layer method to find the substrate concentration gradient. The mass balance at the differential layer is a change of substrate concentration equals to the flux in minus the flux out minus the consumption. At steady state, there's no change of substrate concentration in the differential layer. We can substitute Fick's first law into the mass balance and get this diffusion reaction equation. The boundary conditions for the second order differential equation are: at the substratum, there's no change of substrate concentration as there's no reaction. So dSF over dx equals to 0. At the biofilm surface, the substrate concentration equal to S sub Ss. This is a nonlinear second order differential equation. It does not have an analytical solution. We can solve it numerically. Computer programs makes that a lot easier than solving it by hand. Or, if we want to get a quick estimation by hand, we can use approximation approaches. There are 3 major approximation approaches we can use. I will cover the effectiveness factor approach now in this class and talk about the other 2 in the next class. The effectiveness factor approach basically summarizes the substrate concentration profile into an effectiveness factor, eta E, that modifies the overall growth rate in biofilm and use the bulk solution concentration to calculate the growth kinetics. Eta E should be calculated from the integration of the substrate concentration profile in the biofilm and boundary layer, but its solution can be simplified. We can find the effectiveness factor from this figure after calculating the Thiele modulus and the Biot number. The Thiele modulus squared is a ratio between the maximum possible first-order reaction rate and the maximum possible diffusion rate. We can correct the Thiele modulus for non-first-order reactions with a ratio of S sub Sb over K sub S. When the Thiele modulus is high, it means that the biofilm is diffusion limited. The consumption of substrate is controlled by the diffusion rate. When the Thiele modulus is low, then the biofilm is reaction limited. the substrate consumption is controlled by the microbial growth. The Biot number is a ratio of external mass transfer coefficient at the stagnant boundary layer to the internal mass transfer coefficient in the biofilm. Note that the external mass transfer coefficient is the diffusivity of substrate in water divided by the boundary layer thickness. Because the boundary layer thickness is difficult to determine, so they are summarized into KL. The substrate flux is KL times SSB minus SSS. With the Thiele modulus and the Biot number calculated, we can find the corresponding effectiveness factor from this figure and calculate the flux. Another important note I want to reiterate is that the only driving force for the flux is the concentration gradient. The concentration gradient is created by bioreaction. So, the flux can be described by this reaction term, but also by the concentration gradient in the boundary layer, or by the concentration gradient in the biofilm. With the flux calculated, we can perform a reactor mass balance. The change of substrate concentration in the reactor equals to the mass of substrate in minus the mass of substrate out, minus the flux of substrate into the biofilm, and minus the substrate consumption in the bulk solution by suspended biomass. Let's expand the steady-state reactor mass balance and divide both sides by the volume V. XBHF is the biomass concentration in the biofilm, while XBHB is the biomass concentration in the bulk solution. Biofilm thickness, LF, times the biofilm surface area, AS, is the biofilm volume. We assume that the biofilm biomass has the same mu max and K sub S as the bulk solution biomass. We also assume that the biofilm biomass concentration is an intrinsic property of the biofilm. So only the bulk solution biomass concentration is determined by the substrate concentration and yield. Now, the equation is simplified enough to find the effluent substrate concentration as a function of constants and design parameters, so the reactor performance can be determined. The effectiveness factor approach is a good way to quickly estimate the performance of a attached growth reactor, but has some issues with real-life simulation. The biofilm thickness needs to be predetermined either by estimation or through experiment. The former can be inaccurate, while the latter is not easily done in real-life design process. This approach is easy for one substrate, one biomass, but becomes much more complex for modeling multiple species utilizing multiple nutrients, which is more common in real life. We will talk about other approaches that better deals with these issues in the next class.